Welcome to the video for chapter 6.4. This is one of my favorite sections that we talk about all year. This is one of my favorite things to study. So chapter 6.4 is all about microbial growth and the environments microbes can grow in. So we're going to talk about environmental limits, which means we have to talk about organisms that grow in weird conditions, extreme conditions. So we get to talk about my beloved extremophiles in this section. Okay, so let's start the lecture. Okay, so let's start with some review questions. So review question number 1, how would you classify a bacteria that uses carbon dioxide, CO2, as a carbon source that gains energy from H2S and an inorganic compound? Okay, so is it A, an autotroph and an organotroph, B, an autotroph and a lithotroph, C, a heterotroph and an organotroph, or D, a heterotroph and a lithotroph? Okay, so pause the video, take a second, flip through your notes, go maybe back to that chart in chapter 6.3, and see if you can find the answer. Okay, the answer for this is B, an autotroph and a lithotroph. So let's go through the parts of the question that tells us this answer. Okay, so our first clue is that it uses CO2 as a carbon source. Okay, so there's only one type of organism that uses carbon dioxide as a carbon source, and that's going to be an autotroph. This is going to be things like plants and things that do photosynthesis. Okay, so this is going to help us narrow it down to just A and B. Now, if it had said instead of CO2 like C6H12O6 or glucose as a carbon source, then it's going to be C or D because that means it's getting its sugar from somewhere else, whereas autotrophs are going to make it from the carbon dioxide. Okay, so we're now between A and B, so it's either an organotroph or a lithotroph. So remember, organotroph means that it eats organic compounds, organic material, where lithotroph means it eats rocks. Okay, eats rocks means they're going to be inorganic. Okay, so when it uses hydrogen sulfide to gain energy, that means it's an inorganic compound, which means it's a lithotroph as well as an autotroph. So the answer is B. Okay, review question number 2. The blank growth rate for a species occurs at temperatures that allow all the cell's proteins to work most efficiently as a group. Is the answer A, suboptimal, B, optimal, C, minimum, or D, maximum? Okay, so take a second, pause the video, see if you can find the answer. I will say that this is more of a vocabulary question. We've talked about this a little bit in class, but if you don't remember, that's okay too. So take a second, pause, see if you can find the answer. Okay, the answer for this one is B. It's optimal growth rate. Okay, whenever you see the term optimal, this means it's working great. It's working most efficiently. This is the perfect conditions. Okay, so this is either perfect temperature, perfect nutrient level, things like that. You really don't see minimum and maximum with the growth rate. It's usually just optimal or suboptimal. So suboptimal, if this question was to be rewritten for that, it'll say it would be occurs at temperatures that do not allow the cell's proteins to work as efficiently, or the cell's proteins are going to work inefficiently. That's suboptimal. Okay, so the answer for this one is B. Okay, so review question number 3. Which of the following is not required for microorganisms to grow and divide? A, DNA replication, B, binary fission, C, equatorial septum formation, or D, conjugation. Okay, so pause the video, flip through your notes, and see if you can find the answer. Ooh, sorry. Okay, the answer for this one is conjugation. Now, if you remember from a few sections ago, probably in 6.32, conjugation is when we use the pili. So we're going to use that conjugation pilus to exchange genetic material back and forth. Now, this is going to be either parts of the genome or plasmids. Okay, neither of those are required for the microorganism to grow and divide. Well, DNA replication, so A, is required, right? We have to copy our DNA, or they have to copy their DNA, or they can't be functional. Okay, so if the DNA is not copied before they divide, one of the daughter cells is not going to have a genome, so it's not going to be able to function. Okay, binary fission, this is actually the process of dividing, so this is definitely required. Equatorial septum formation, this one's thrown in there to kind of throw you a curveball. Okay, so don't mind this equatorial part. This just means it's around the middle, just like the equator is around the center of the earth. Okay, what's important here is septum formation. Remember, this is a part of binary fission. So if the septum doesn't form, the parent cell is not going to divide into the daughter cells. Okay, so the answer is D, conjugation. Okay, so 6.4, this is all about environmental limits on microbial growth. So we're going to talk about some really interesting bacteria and archaea in this section. They're really cool. I love them so much. Okay, so our section objectives are describe how environmental changes can alter the shape of a growth curve. You need to be able to list different classes of microbes based on their preferred environmental niches. So this is things like pH, temperature, and salt content. You need to be able to identify the biological properties that allow different classes of microbes to grow in extreme environments. Okay, so what makes them survive in these weird places? Okay, so we're going to talk about these. I'm going to try not to ramble too much because I do love this topic so much. This is what I studied when I did microbiology. Okay, so let's start. Okay, so one of the things that is really cool about bacteria is they are a very diverse group of organisms. Okay, not only are they physiologically diverse, so we have different shapes, right? We have bacilli, we have cocci, we have spirochetes, we have different arrangements. Okay, but they're also metabolically different. Okay, so they have different chemical reactions they do. They make their energy different ways. They get food different ways. Okay, so in addition to the nutrients that we talked about in 6.3, we need to talk about other things that we have to consider when growing bacteria. Okay, so this is going to be the temperature and pressure of the environment, something called osmotic balance. Okay, aka how much salt is there in the environment? Okay, lots of salt. A lot of organisms don't like lots of salt in their environment. That's bad growing conditions. Okay, so osmotic balance, this is related to tonicity. So hypertonic, hypotonic, isotonic. Okay, so think about that. So think about how much salt you have in that environment. And then finally, pH level. Okay, so this next part is kind of subjective. What really is normal anyway? For us, we define normal conditions as where we grow at. Okay, so normal conditions have a temperature between 20 degrees C and 40 degrees C. This is like 68 degrees Fahrenheit and 104 degrees Fahrenheit. Near neutral pH, so something like 6.8, 7.2. So right around that 7 mark on the pH scale. A low salt concentration of around 0.9%. So very, very light on the salt in normal conditions. And normal conditions are going to have enough nutrients to support life. Okay, so this is normal. Okay, we fit into this normal category right here. Now, anything that lives outside these temperature ranges, these pH ranges, these salt concentrations, they have a specific name that we call them. They are what we call extremophiles. Okay, so extremophiles are bacteria, archaea, and some eukaryotic microbes that grow in extreme environments outside of these normal conditions. And we're going to talk about some of them. So let's go to the next slide where we're going to talk about them. Okay, now you're not going to have to know all of these, but these are different environments that bacteria can live in. So really cool ones are polar ice caps, are glaciers, the sea ice, the permafrost. These all have extremophiles. They have little microbes living there. We've talked about this one a little bit. Okay, so we have some things that grow in the ocean. Okay, we could have by the deep sea hydrothermal vents. So these are the sulfur vents. Okay, so sulfur gas is being released, heat's being released. We have the cold seeps and mud volcanoes underneath. We have shallow water thermal vents. We have deep sea lakes. Okay, this is usually going to be something that has a temperature change, and it's going to have a higher salt concentration, which is why it's going to form a lake on, say, the bottom of the ocean where you wouldn't expect to see a lake. Okay, so with the cold seeps, the mud volcanoes, they're all going to have interesting nutrient levels that are going to be unique to the extremophiles that live there. Okay, we have serpentinizing environments. Okay, so we have some different rocks, some different cracks that they can live in. Okay, we can have them living in what we call the continental subsurface of the ocean, so in the dirt that makes up the ocean floor. Okay, so that happens at different levels. Could be clear at the bottom of a trench. This could be near where the beach is. One of my favorites, okay, Yellowstone. This is where Yellowstone fits in. Okay, these are going to be hot springs, fumaroles, and mud volcanoes that are above the ocean level. We have acid lakes and volcanoes. Yes, there are things that grow in a volcano. Isn't that cool? We have different organisms that live in arid or dry conditions like deserts. Um, we have hyperacidic lakes, but we also have what are called soda lakes. These are basic lakes, so highly basic. They're sometimes called alkaline lakes. We also have super hypersaline lakes, so this is going to be stuff like the Great Salt Lake and the Dead Sea, where they have really high salt contents. We have, let me see if I can say this correctly. I did ask Mrs. Taylor before we started doing this. Ophiolites and continental serpentinization. There is literally extremophiles that live inside of rocks. How cool is that? In fact, Mrs. Taylor has given me a rock sample to show you guys where we can see where those extremophiles will be living. Okay, and some of the weirder ones that kind of make sense if you think about them would be like Chernobyl, our nuclear contamination sites, where we've had some nuclear meltdowns. We have special extremophiles that have learned how to live in those environments where there's radiation in the soil. And so one of the things that some mining practices do is they use super acidic water to cut through the mountains or into the ground. And because of that, we get this acidic drainage by the mines. We have bacteria that have evolved and adapted to live in the mine drainage, which is so crazy to me. Okay, so there's more extremophiles than just these. These are kind of the cream of the crop, the ones that we know the most about. Okay, but there are a few more extreme conditions where we can find extremophiles. Okay, I would know a couple of these for the exam. Okay, so do not panic. Okay, let's talk about what you do need to know for the exam. Know some places where you can find these extremophiles, but also we need to know some terminology for the test that concerns these extremophiles. Okay, we need to be able to classify them by where they live and the conditions they can handle or prefer. Now, I'm going to say this. You do not have to memorize the numbers. You don't have to know the ranges. You have to just kind of have a basic understanding of what these terms mean. Okay, so you're going to be tested on the temperature extremophiles. Okay, we have hyperthermophiles, thermophiles, mesophiles, and psychrophiles. Okay, so hyperthermophiles, these are going to live at really extremely hot conditions. So inside volcanoes, by the hydrothermal vents on the ocean floor. Okay, so they're going to live in areas that are above 100 or 176 degrees Fahrenheit or 80 degrees Celsius. Okay, the opposite of the spectrum, we have the psychrophiles. Okay, these are going to live in conditions that are below 15 degrees Celsius, so like below 60 degrees Fahrenheit. We have the mesophiles. Okay, these are like us, like a lot of human pathogens are mesophiles. They live between that 15 degrees C and 45 degrees C range, so 60 degrees Fahrenheit to about 115 degrees Fahrenheit. Okay, and then the thermophiles, these are the things that grow up in Yellowstone in the hot pots, that they can live in a range between 50 degrees C and 80 degrees C, so 122 to 176. Okay, so that's the temperature conditions. So another condition that we look for is the pH. Are they in a neutral condition? Are they in an acidic environment, or are they in a basic environment? Okay, so if the organism's living in a neutral environment, This means that they are a neutrophile. We are neutrophiles, by the way. They live in pHs from 5 to 8. Okay, so we have the hyperacidic lakes, so the areas of high acidity. Okay, so things like our stomach acid would be highly acidic. Okay, organisms that can live there are what we call acidophiles. Okay, they're going to live in pHs below 3. And we are going to talk about an acidophile that actually can cause disease in you and me. Okay, and then we have the organisms that can live in basic environments like those alkaline lakes. These are the alkalophiles. Whenever you see alkali, that means basic. So like group 1 on the periodic table is the alkali metals. They make a lot of bases, which is why they're called the alkali metals. Okay, so alkalophiles are going to be above a pH of 9. Okay, so for osmolarity, so that salt content, we really just have one that we're going to talk about, and these are called the halophiles. This is because almost all salts have a halogen, so an element from group 17. So halo means salt, halogen means salt. Okay, so halophiles means that they're going to live in high salt concentrations, like really extremely salty. Okay, so organisms that live in the Great Salt Lake, these are going to be halophiles. Okay, so we have another condition. How much oxygen do we have there? We say aerobic and anaerobic conditions. We're going to talk more about this oxygen environmental limit in 6.5. So we'll talk more about that. But we can have strict aerobes, facultative microbes, okay, microaerophiles, and strict anaerobes. Sorry, guys. But we'll talk more about that in chapter 6.5. Okay, and then finally, we have those extreme pressure extremophiles. Okay, so we have barotolerant. This will be anywhere from 10 to 500 atmospheres. Whereas we have the baryophiles. These are going to be organisms found at high pressure conditions. Okay, so greater than 380 atmospheres. Baryophiles are going to live places like at the bottom of the ocean. Like we can't go to the bottom of the ocean because of the extreme pressure, because of all the water and the air above us. So we can't go down there. We can go down in submersibles. We have problems diving deep because of not just the oxygen, but the different gases that are dissolved in our blood. Because if we go down really far and then come up really quickly, gases are going to so as you go down in pressure, they're going to dissolve into your bloodstream better. But as you come back up, they are going to start to expand out of your bloodstream, which then can cause things called a condition called the bends, which literally kind of tears your guts up from the expanding gases in your intestinal tract. Not a good place to be. There's a reason why when we fly or when we go into submersibles that they are pressurized to avoid that kind of stuff. Okay, so these are the different environmental conditions. As I said, don't know the numbers, know the names, know what they mean, but we're going to talk about these in some more detail here. Okay, so let's talk about our thermophiles, the ones that go through the different temperature conditions. Now, one of the things I want you to notice here in this graph is that these different extremophiles are going to overlap a little bit. Okay, so psychrophiles are going to go from about 0 to 20 degrees C. Mesophiles are about 12 to 55 degrees C. Thermophiles are from about 42 to 68 degrees Celsius, while hyperthermophiles are about 64 to 110, 115 degrees C. Okay, so there's always going to be these areas of overlap where you can find both. Okay, so that's important to know, but we need to know these in order of increasing temperature. So psychrophiles are our coldest, mesophiles are the next class up, thermophiles, they can live in really hot conditions, and hyperthermophiles can live in extremely hot conditions. Okay, so let's talk about mesophiles, because me and you, we function best at about 37 degrees Celsius. The organisms that live at that temperature are what are called mesophiles. So our lab strains of E. coli and our lab strain of Staphylococcus epidermidis that are in the back of the classroom right now, these are all mesophiles because I prefer that 37 degrees Fahrenheit. Most human pathogens, so things that cause disease, are also going to be mesophiles because that optimal growth range is between 20 degrees C and 40 degrees C. Okay, this is important. We need to know this mesophiles because we're going to come back to this term quite a bit throughout the rest of the year, well, not the year, the rest of the semester, as we talk about how things cause disease. Okay, so let's talk about the ones that do not live at normal temperatures, like the mesophiles. So we have the thermophiles. These are going to be bacteria and archaea that can live at high temperatures. So think about 130 degrees Fahrenheit or higher. I like to think about Yellowstone. So Grand Prismatic, which is one of the most not important, but one of the most famous features in Yellowstone, has all those really pretty yellows, oranges, greens, reds. Those are all caused by different thermophiles, which is pretty cool. Okay, so we have our picture of Yellowstone here from the textbook. Okay, so we see these greens and these yellows, and we see the kind of like cyan color. These are all different microorganisms that we call thermophiles. Okay, so Yellowstone's hot, but it's not the hottest we can get here on the planet. This is where we have hypothermophiles. Okay, so hyperthermophiles are going to grow at temperatures that are above 250 degrees Fahrenheit or 121 degrees Celsius. To get to these temperatures, though, outside of a volcano, we need to have extreme pressure, just like we have to use autoclaves to get water above that boiling 100 degrees Celsius temperature to kill things like endospores. The same thing happens at the bottom of the ocean. So we're going to have that extreme pressure. Okay, and we're going to have the really hot thermal vents. So this is where it's going to be that hyperthermophilic condition. Okay, so high pressure, high temperature. So hyperthermophiles are also a lot of time baryophiles. Okay, they are going to be able to live at high pressures. Okay, the really important thing to remember is that all of these extremophiles are going to have specialized structures that allow them to live in these extreme conditions. So thermophiles themselves have cell membranes and proteins that have adapted to high temperatures. So we can have more cholesterol found in the cell membrane. We talked about this in biology about how the cell membrane is going to keep membranes fluid at cold temperatures and more rigid at high temperatures, so they don't get so fluid they actually just dissipate and flow away. Okay, so thermophiles, if we took their proteins and put them at a mesophilic condition, they're not going to work. That's not their optimal temperature. Just the same as if we took our proteins and put them at the same temperatures thermophiles live at, it's actually going to denature our proteins and make it so that they don't work. They're going to be suboptimal conditions. Okay, so this is an important thing to remember because a lot of scientists make their careers out of studying weird stuff. Okay, so one such scientist is a scientist by the name of Thomas Brock. Okay, so Thomas Brock is pretty famous. He loves studying weird things, but he's also really the first scientist that realized that there were things growing in the hot pots at Yellowstone. Okay, so he made it his career to study the things in the hot pots up at Yellowstone. Okay, so he is really famous for discovering a thermophilic bacteria that lives up in Yellowstone. He got it from the mushroom pool. I've only been here once. This is an area that's kind of close to, well, it is close to tourists, but they don't want a lot of people back there because this thermophilic bacteria lives there. Okay, so Thomas Brock named it Thermus aquaticus. Thermus literally means hot. Aquaticus means water. So this bacteria is named hot water. Okay, because it lives in hot water. Okay, so Thomas Brock identified it. He studied it. He collected some samples and took them back to the lab. But the thing is, when he was done studying it, he kind of just stored it away. So usually when we're storing things like bacteria to keep them for another day. We're going to put them in a really cold freezer called a minus 80 degrees or minus 80 freezer. So that's where he put these bacteria. Now, when Brock discovered Thermus aquaticus in 1969, we had barely figured out what the shape of DNA was. We were still figuring out how DNA copied itself. Like, there was a lot of unknowns at this time. So as work happened with DNA and we started to learn more and understand the process of DNA replication, a lot of scientists started to wonder if we could take an organism's DNA and if we could replicate it in a test tube. Now, these people weren't very successful at first, because they kept trying to use human proteins. And the thing is, when you replicate DNA in a test tube, in order to get the two strands that make up DNA to separate, we're going to have to do we have to heat it up. Okay, we have to heat it up so much that the hydrogen bonds between the two strands break, and whatever enzyme we put in there can get in there and replicate it. But the problem is that every time they tried to do this, our human proteins weren't good enough. When they increased the temperature to separate the strands, we would also denature our proteins. So it wasn't working. So in the 80s, there was a scientist by the name of Kary Mullis. He's the first one that kind of took Thomas Brock's work and started to connect it with this idea of replicating DNA in a test tube. Okay, he's the one that made the connection that these bacteria, so Thermus aquaticus, can survive in hot springs, which means that they have enzymes that do their DNA replication that can be heated to high temperatures and still function, unlike ours. Okay, so he contacted Thomas Brock, and Thomas Brock went and got his sample of Thermus aquaticus out of the freezer and handed it to Kary Mullis. So Kary Mullis's research group looked at Thermus aquaticus and looked at its process of replicating its genome. And what they did is they were able to find an enzyme called tag polymerase. Okay, and it could survive at these high temperatures, and it could help replicate DNA in a test tube. Okay, so with the discovery of tag polymerase, a new technology by the name of polymerase chain reaction, or PCR, we did this in biology 201, remember? Okay, this is when it was developed, and we started using it, is when Kary Mullis made this connection in the 80s. Okay, in fact, without the discovery of tag polymerase and using it for PCR, we wouldn't have had the Human Genome Project that started in the early 90s and went to the early 2000s, where we sequenced the genome of a human for the first time. Because what PCR does is it's going to copy these strands of DNA and amplify them. So we get a lot so we can actually look at them. So without being able to amplify DNA, we wouldn't have been able to sequence the human genome. Okay, so tag polymerase was able to step in where our enzymes and our proteins could not. So one of the things that came out of this application of Thermus aquaticus and the discovery of tag polymerase is that we learned that biodiversity, so having those extremophiles, could lead to making a lot of money. So Kary Mullis, with his invention of PCR and the use of tag polymerase, he patented that and has made billions of dollars since then. Now, you have to remember, Thomas Brock found Thermus aquaticus in Yellowstone. Okay, but the thing is, usually when you're doing research on federal lands, such as in the national park, they usually have you sign an agreement now that states that if you find anything or discover anything that is going to bring profit, you're going to share some of those profits with the national parks. But the thing is, when Thomas Brock isolated Thermus aquaticus first and discovered it in Yellowstone and studied it, there wasn't any way that they could think that there would be a commercial application for it, because Thomas Brock was just studying it to learn more things and just gain knowledge. Okay, no commercial application was seen at the time. Okay, so when Carey Mullis went and patented tag polymerase as used for PCR and started to make all this money, he didn't have to give any to the National Park Service because there's no agreement in place. It was a big to-do. The National Park Service understandably felt very cheated out of a lot of money. I do believe that Wallace's company that developed PCR has since given a bunch of money to Yellowstone to kind of make amends for making all this money off of Yellowstone. But now, because even though we can't see a commercial application for things that we're studying in the national parks, all researchers that go into the national park to conduct research have to sign an agreement with the National Park Service saying that we have to share profits if we discover anything that turns a profit. So, big business getting in the way of science yet again. I've done some research in national parks. I've signed these agreements. They are pretty ironclad, actually. Okay, so we've talked about mesophiles, thermophiles, hyperthermophiles. We've talked about Thermos aquaticus. Let's talk about the last temperature range, which is the really cold guys. So the guys that live in really cold conditions. Okay, so these are the psychrophiles. Okay, so they can grow in temperatures as low as negative 10 degrees Celsius or 14 degrees Fahrenheit. Okay, so a lot of times when we're saving things, so like for our lab, so for our class, we have a bunch of standards, a bunch of what we call slants of bacteria that we use to do our experiments with. I keep them in the fridge between me and Mrs. Taylor's classroom at about 32 degrees Fahrenheit, and they don't grow. They just stay dormant. Okay, so the fact that psychrophiles can grow at even colder conditions than the refrigerator that we keep our slants in is kind of crazy. Okay, but one of the ways that psychophilic microbes have evolved to survive in cold temperatures, because normally organisms that get put in those cold conditions that aren't used to them, their cell membranes are going to get very rigid and they're not going to be able to move and they're going to get very fragile and like crack and break because of the cold. So psychophilic microbes are going to have more proteins that are flexible and more cholesterol to keep their phospholipid bilayer, about blanked guess, their phospholipid bilayer fluid. Okay, so they're going to be specifically adapted to these conditions, which is kind of cool. So no psychophilic is the coldest, hyperthermophile the warmest, mesophile and thermophile are in between. And know where you can find these guys. Okay, so the last one, well, not the last one, so the next one we're going to talk about is those osmotic environments, those osmolarity. So osmolarity is defined as the number of solute molecules dissolved in solution. Okay, so the more solute molecules dissolved means the higher the osmolarity. Okay, so when we're talking about osmolarity, when it comes to extremophiles and conditions microbes can live in, we're talking about salt. Okay, how much water is available, how much salt is in that water. Oh, excuse me. Okay, so there is specifically some species of archaea that we call halophiles that have evolved to live in areas with high concentrations of salt. Okay, so they are going to actually have in their cells, they're going to have what are called osmolytes. Okay, so it's going to make inside of their cells more concentrated than the outside environment. Okay, so it's going to be a hypotonic condition where they're going to have a greater concentration inside than outside, which means that all their water is not going to rush out when they're in a water environment, like it would when we've talked about hypertonic conditions, where there's more salt concentration outside than inside the cell, so all the water rushes out to equalize that. Okay, to prevent that, halophiles are going to have these salts that are dissolved inside their cells and have higher osmolytes than regular organisms, so that they don't have this, so that they can actually bring water in and reverse what normally happens. Okay, so halophiles love salt. Okay, that's literally what halophile means. One of the ways we used to preserve food and make sure it would last before before refrigeration, was that we used to cure things in salt and pack things in salt. Okay, so salt has been great for keeping things are keeping things from growing, so limiting microbial growth, because a lot of microbes cannot live in those high salt conditions because it makes all of the water leave their bodies, or, well, their cells, not their bodies. Okay, so normal microbes don't like high salt. Okay, so a lot of times when you see halophiles, like at the Great Salt Lake, or in these salt flats, you're going to see this pink color. Okay, so this pink color means that's where those extremophiles are found. Okay, so the next conditions that we're going to talk about are the pH conditions. So the things that live in acids, bases, and in between. Okay, so a lot of things that we talk about that affect us as humans are going to be neutralophiles. They're going to live in this range of a pH of 5 to 8. Okay, where acidophiles are going to live outside of that normal range. So let's talk about these different types. Okay, as I said, neutrophils, they typically grow between pH of 5 and a pH of 8. So all human pathogens are going to be neutrophils. So human pathogens are neutrophils and mesophiles. Important things to remember. Okay, so acidophiles are going to be the bacteria and archaea that live in acidic conditions and acidic environments. We actually have a pathogen that infects humans. It's called Helicobacter pylori. Okay, so Helicobacter pylori lives in your stomach. It lives in the stomach acid, and it causes ulcers. Okay, so one of the reasons why Helicobacter pylori can live in such an acidic condition is it's going to create a molecule called urease. Okay, it's very similar to urea that makes up urine. So urease is basic. Okay, it contains a lot of ammonia molecules. So Helicobacter pylori is going to release this urease, these ammonia ions, in the stomach acid. So it's releasing a base, it's interacting with the acid, so we're going to get a neutralization reaction happening. Okay, so Helicobacter pylori is basically making itself a neutral bubble around it so it can live in the stomach. Okay, so the damage that it causes by releasing urease and these neutralization reactions are going to actually cause the ulcers to form. Okay, so that's acidophiles. Alkalophiles are going to live in the opposite end of the pH spectrum. They're going to be from a pH of 9 to about 12. Okay, so we find them a lot in alkaline lakes. Okay, so lakes that are basic. One of our common alkalophiles is this spirochete. Okay, this curly fried looking dude. This is spirulina. Okay, spirulina looks green, but you see this alkali lake here in the picture. Now it's pink. Spirulina, even though it looks green, produces a molecule or a pigment that is pink, which is why when they're in large concentrations, like in this alkali lake, you can see it as pink. So it turns pink. Okay, so there's lots of organisms that eat spirulina. Okay, so things like fish and bugs and other small microorganisms. So what's really funny is flamingos, they eat spirulina. Well, okay, they eat things that eat spirulina. Because of this, because they're eating spirulina, even indirectly, and spirulina releases this pink pigment, this is why flamingos are pink. It's because they're eating spirulina. So, I don't know if you guys know this, but flamingos, when they're born, are white. They're not pink. So they gain their pink color as they start to eat these alkalophiles that are being eaten by their food source. Okay, so this is what a baby flamingo looks like. See, it's white. It's not pink yet. So this little guy is going to turn this color by eating the spirulina, which is kind of fun. Okay, so now we know why flamingos are turned pink. So it's also why we have such variation in the color of pink that flamingos can be, is because there's different concentration of spirulina in their food. So like a lot of zoos, their flamingos are going to be a little bit less pink because they don't have naturally occurring alkaline lakes in the zoos. So they're going to import things that have eaten spirulina to keep them pink, but since they're importing it, it's not going to be, the flamingos in the zoo aren't going to be as pink as the ones that are out in the wild. Okay, so one of the things that we need to talk about has to be with these growing conditions, these environmental limits. Okay, so whether it's a normal microbe that lives in the normal conditions or one of our extremophiles, all bacteria are going to sense their environment. Okay, so they're going to make decisions based on what they're sensing. So bacteria, when they're sensing their environment, can actually turn genes on and turn genes off, so they can start making proteins that are going to help them be more successful in that new environment, depending on what they're sensing. Okay, so pathogens do this when they enter a host. Okay, so let's look at an example about this. Yeah, so we have Salmonella, Enterica, Cerevar, Typhimurium. Oh, this is a hard one for you guys. Typhimurium. Okay, I'm just going to say Salmonella. Okay, so this strain of Salmonella causes a lot of gut disease, so gastrointestinal problems, a lot of bloody diarrhea, because that seems to be a thing with most bacteria that infect your digestive tract. Okay, so the thing is, Salmonella enterica, no matter the strain, is usually enters the host by the oral route. Okay, so you're going to ingest contaminated food, and that's how you're going to get salmonella. Okay, so what happens is, as you eat the food that's contaminated with salmonella, you're going to chew up the food, it's going to go into your throat, and the first stop it's going to make is in your stomach. Okay, so it's going to start to sense that it's in an acidic environment, which is going to flip the switch, and it's going to start to make a bunch of different proteins that are going to allow it to survive once it gets out of the stomach. Okay, so as it gets out of the stomach, it's going to get into the intestinal tract. The intestinal tract is a little bit basic. Okay, so these different proteins that it's going to start making once it senses that change in environment is going to be what makes it successful in infecting our intestines. Okay, and so some of these proteins that Salmonella makes are going to allow that bacteria to trick the macrophages, the neutrophils, to be more specific, our white blood cells, into eating them by phagocytosis. But the thing is, these proteins, once they are phagocytosed, so they come into the cell, once they're in the cell, these proteins are going to manifest themselves on the outside of the endosome. Okay, so it's going to tell it not to merge that endosome with the lysosome, so they're not going to get digested with the lysosome. They're actually going to now have a nice little home that allows them to hide from the immune system and be in a good environment where they have plenty of nutrients, where they can grow and divide and cause infection and cause disease. Kind of cool, kind of scary, but all pathogens can sense this change when they enter the host. Okay, and they're going to trigger different proteins depending on where they're at. This is also why different pathogens are going to have that tissue tropism, so they're going to infect different areas of the body, because different areas of the body are going to sense differently and are going to trigger a different set of proteins. Okay, so the ones that they're most likely to infect, so like Salmonella enterica in the guts, this is going to be because their proteins are beautifully evolved to make them to help them, not make them, help them survive in that environment. Okay, so that is section 6.4. So we talked about the different extremophiles. We talked about where they live, their environmental niches. Okay, we've talked about how they sense their environment. Okay, so in 6.5, we are going to finish up this talk about extremophiles, but we're going to talk about the no oxygen conditions for those extremophiles. So we will see you in section 6.5.